Today I'll talk about something very interesting. So any animal with a lot of cells that undergo a lot of cell division should logically experience a higher rate of genetic mutation, leading to a higher risk of cancer, right? Because there's a higher risk of the mutation occurring in an oncogene or a tumor suppressor gene leading to cancer. But then why do elephants who have 100 times more number of cells than humans rarely get cancer? So this is because of something called the Peto's paradox, which means massive long-lived animals get cancer at a fraction of the rate of smaller animals. So how does this happen in elephants? One, because of the TP53 gene. The TP53 gene is known as the guardian of the genome. Its function is to protect your body from cancer by regulating cell division, repairing damaged DNA, and destroying cells with irreversible mutations. Humans have 2 genes of TP53, but elephants have 40 genes of TP53. So if an elephant gene mutates, it has 20 times more genetic power to detect it, to repair it. And because elephants have so many different variants of the TP53 gene, their cells can recognize a much wider spectrum of molecular triggers and DNA damage than human cells. Second reason, LIF6. This is the leukemia inhibitory factor 6 gene, and this is actually a zombie gene because it has been dead for years of evolution, but it's there in elephants as an extra gene and it works as a potent tumor killer. So when an elephant cell gets damaged or miscopied, the TP53 genes act like an alarm system and they immediately awaken the LIF6 gene, which goes straight into the mutated cell and forces it to kill itself so it can never become a cancerous cell. And the third reason is that human cells usually try to repair damage done by radiation to the DNA, but elephant cells do not do that. So when elephant cells were blasted with radiation in the lab, they committed cellular suicide at twice the rate of normal human cells because they prioritized the death of a damaged cell to protect the rest of the body. So I spoke about the Peto's paradox, which is how large animals, even though they have more number of cells, more cell division, more chances of mutation, and more chances of mutation occurring in the oncogene or tumor suppressor genes, still get cancer extremely rarely. This is the Peto's paradox, and I spoke about elephants in particular. But are there other animals also that get cancer very rarely, extremely rarely, and why? So elephants have 100 times more cells than humans and they get cancer rarely. But whales have 1,000 times more cells than humans and even they rarely get cancer. So the mechanism in whales is, instead of duplicating tumor suppressor genes, whales rely on superior DNA repair pathways. So their cells overexpress repair proteins like ERCC1, allowing them to fix double-stranded DNA breaks with a much higher accuracy and efficiency than human cells. And apart from whales, apart from larger animals, there are also small animals that get cancer very rarely. So one example is the blind mole rat. So these are subterranean rodents and live up to 21 years but do not develop tumors. And they have evolved a targeted continuous immune response that rapidly locates and induces early cell death called apoptosis in precancerous cells. Because they live in low oxygen environments, their metabolic pathways are structurally altered to naturally suppress rogue cell growth. And another animal is the naked mole rat. So these are uniquely resistant to both spontaneously and chemically induced tumors, and they live up to 30 years. The mechanism is that they secrete high concentrations of a thick sugar-like molecule called hyaluron in long chains, HMM/HA. When a mutating cell begins to crowd or divide rapidly, hyaluron triggers early contact inhibition, stopping cell proliferation before a tumor can form. And this is how some animals get cancer extremely rarely, and maybe some research can be done to mimic these mechanisms. I have spoken about cancers in certain animals, or how some animals are cancer resistant and some animals do get cancer just like humans do. The frequency is the same. Today I will talk about a cancer in an animal that is transmissible. Now this is very rare, very lethal, but it does happen. And it happens in the Tasmanian devil, and it's known as the Tasmanian devil facial tumor disease, DFTD. So, what happens here is that these Tasmanian devils are naturally aggressive and bite each other's faces during mating and feeding. So, when an infected devil bites a healthy one, living cancer cells are physically scraped off and grafted directly into the bite wounds. And normally, an animal's immune system immediately rejects and kills any tissue from another individual. But DFTD cells completely turn off their MHC identity tags. So MHC is major histocompatibility complex, which helps the immune cells see cancer cells, and this is turned off in DFTD cells. So therefore they become invisible and the new host's immune system ignores them. The tumors can grow into massive horrific lumps all over the face, jaw and inside the mouth. The animals die within months because the physical size of the tumors completely blocks them from breaking down food, leading to starvation. Now, where did this originate from? So, there are two strains of DFTD. One is DFT1, which was first identified in 1996 and is believed to have originated from a Schwann cell, that means a nerve supporting cell, in a female Tasmanian devil. It spread across 90% of Tasmania. And the second strain is DFT2. This was detected in 2014 and originated from a male Tasmanian devil. And fortunately, DFT2 appears to be geographically confined to a peninsula in southeastern Tasmania. So I spoke about a transmissible cancer in the Tasmanian devil, DFTD, the Tasmanian devil facial tumor disease. Now I'll talk about another transmissible cancer that is seen in dogs. This is called canine transmissible venereal tumor, CTVT, also called Sticker's sarcoma. This transmissible cancer, CTVT, is called a living allograft. An allograft is just a tissue transfer between an individual of the same species. So normally a transplant requires a doctor to surgically remove a healthy organ. But CTVT is called a living allograft because the cancer cells act like a transplant that moves by itself. So when two dogs mate, a clump of living cells physically detaches from one dog and implants itself onto the other dog. It stays alive and continues to grow. And here the host dog's DNA does not match the tumor DNA. Why? Because in a normal cancer, like let's say a human breast cancer or lung cancer, a person's own cells mutate. So if you test that tumor, the DNA will match the patient's DNA because the cancer grew from their own body. But CTVT does not work that way, because the tumor cells are actually the living tissue of that founder dog from 11,000 years ago where it originated from. The tumor has its own ancient DNA. The mode of transmission of CTVT can be one, physical implantation, so the disease spreads by physical transfer of intact viable cancer cells. Two, friction and injury happens during mating. The natural friction during mating causes some abrasions in the tissue, letting fragile, easily detached cancer cells embed into the new host. And three, non-venereal spread. So cells can transition via social behaviors like licking, sniffing, biting. This gives rise to secondary tumors in the mouth or nasal cavities. And how does CTVT evade the immune system? Three main reasons. MHC downregulation. So just like the DFTD in Tasmanian devils, a dog's normal immune system would immediately reject tissue transplants, but CTVT evades this by suppressing major histocompatibility complex MHC molecules, so making the tumor cells invisible to immune cells. Two, immunosuppressive secretions. So the tumor would actively secrete some things like TGF beta that can paralyze the local immune cells trying to attack the tumor. And three, stealing the host energy. So to prevent genetic decay over years, CTVT steals healthy mitochondrial DNA from its host cells to patch its own damaged metabolic systems. Cunning. CTVT follows a triphasic growth pattern. So the tumor uniquely progresses through three phases. One, a progressive growth phase, two, a static plateau phase, and third, a natural immune regression phase where the host's immune system overrides the evasion and shrinks the tumor. And one good thing is that this is highly treatable. So despite thousands of years of survival, this cancer is responsive to a single agent chemotherapy using vincristine, which routinely achieves complete clinical remission. And it has a genetic fingerprint. So every single CTVT tumor worldwide shares a specific genetic signature, which is a one-line element insertion near the c-Myc oncogene, which drives continuous cell division. So I spoke about transmissible cancers in Tasmanian devils and dogs. Can humans also get transmissible cancers? So naturally occurring large-scale transmissible cancers like the ones in dogs and Tasmanian devils do not exist in humans. But accidental human-to-human transmission is possible under highly specific extreme conditions. So when and how? Human-to-human cancer transmission can only happen in 4 rare accidental settings. 1. Organ transplantation. An organ recipient unknowingly receives a kidney, liver or heart from a deceased donor who had undetected cancer. 2. Pregnancy, mother to fetus. Highly aggressive cancer cells like melanoma or leukemia cross the placental barrier from a pregnant mother into her unborn baby. 3. Surgical and lab accidents. So a surgeon accidentally cuts their hand while removing a patient's tumor, or a lab researcher accidentally pricks themselves with a needle loaded with live human cancer cells. 4. Cross-species infection, the exception. In an extraordinary case published in the New England Journal of Medicine, an HIV/AIDS patient with a severely destroyed immune system accidentally inhaled cells from a common dwarf tapeworm. The tapeworm's cancer cells multiplied inside the human body. But for a foreign cancer to survive in a human host, it must exploit 4 biological pathways. 1. Immune suppression. The host's immune system must be non-functional. Transplant patients take heavy immunosuppressive drugs so their bodies don't reject the organ that they're receiving. And this allows the hitchhiking cancer cells to grow freely without being attacked. 2. Placental privilege. The placenta naturally dampens the mother's immune response, so her body does not reject the baby. Aggressive cancer cells exploit this blind spot to sneak into the fetal bloodstream. 3. HLA allele deletion. In mother-to-fetus transmission, the tumor cells delete human leukocyte antigen HLA genes. HLA genes act like ID badges. So by deleting them, the cancer cells become invisible to the baby's immune system. And 4. Massive cell payload. In surgical or needle accidents, a direct high volume injection of live cells into deep tissue bypasses the skin barrier, creating a temporary local pocket where the cells can attempt to graft.